Welcome to our final session dealing with two central processes of cell division, mitosis and meiosis. So, what are the differences between mitosis and meiosis? Overall, few things are basically the same. Both processes produce new cells. They have similar basic steps and start with a single parent cells. However, mitosis has 4 stages in total, plus the interphase. In meiosis, 8 stages play a role, and we are going to be looking at those in the following few minutes. Mitosis happens in somatic cells. In contrast, meiosis happens in sex or germ cells. The purpose of mitosis is cellular proliferation. Meiosis has the purpose of sexual reproduction. The result of mitosis are 2 diploid daughter cells. In contrast, the result of meiosis are 4 haploid daughter cells having just half the number of chromosomes. In mitosis, usually the genetic variation doesn't change. This is very well the case in meiosis, as we will see in few minutes' time. So again, the result of meiosis are 4 daughter cells, and each daughter cell has just half of the chromosomes of the parent cell. So the result are haploid daughter cells with the chromosomal configuration of 1n. In contrast to mitosis, meiosis can be divided into 2 sets of cell division. But why do we need haploid cells at all? The simple answer is because fertilization combines the genetic material of the father with the genetic material of the mother. So we need cells that have just half the number of the chromosomes. Otherwise, we would end up with a chromosomal configuration of 4n. And this makes absolutely sense, because both the egg cell must have 23 chromosomes or 1n configuration, and the sperm needs the same, because the result of fusion of those 2 cells or result of the fertilization is a cell which should have 46 chromosomes and the chromosomal configuration of 2n. So the purpose of meiosis is to reduce the chromosome number in the sex cells to 23. And this also ensures that every gamete is genetically unique. Meiosis also ensures recombination, and this makes diversity. Diversity is actually very, very important, not necessarily at the individual level, but at the level of population. This is quite evident if you think, for example, about the immune system. Humans are exposed to a variety of new and old disorders, and diversity actually ensures that some of the individuals within a population might already have immunity to a newly occurring disorder. So meiosis scrambles the specific forms of each gene that each sex cell receives. This makes a lot of genetic diversity. This is accomplished via independent assortment and crossing over. And those are processes we are going to be looking at in this section as well. Right, so meiosis is used only for the production of sperm and eggs. It consists of 2 successive cell divisions producing 4 daughter cells. And the first step or first cycle of division during meiosis is usually referred to as meiosis 1. There is also obviously meiosis 2, and the results of those 2 meiotic cell divisions are slightly different. The result of meiosis 1 are actually cells that are still diploid. Haploid cells are only achieved via the second phase of meiosis, meiosis 2. So, let us have a brief look at the different stages of meiosis. You might notice that there is a pictogram showing that this is additional information, and indeed you don't have to memorize every single step for the exam. So during the interphase preceding the meiosis, each of the chromosomes replicates. This results in two genetically identical sister chromatids which remained attached at the centromeres. During the prophase 1 of the meiosis, which is a crucial phase for the mitosis as well, during this phase, each pair of chromatids don't move to the equator alone, they match up with a homologous pair and fasten together in a group of 4 called a tetrad. It is during this phase that crossing over can occur, because homologous chromosomes are actually quite close to each other, and genetic material can be swapped between those chromosomes, and we call this process crossing over. This is the exchange of segments. During this phase, exchange of parts of chromatids follows chromosome pairing. So what you can see here are homolog chromosomes that are close to each other, and those pieces of the chromosomes can be exchanged because the sequence is fairly similar. In the metaphase once, where this exchange already took place, the chromosomes line up at the equator of the cell and they are attached by the centromeres to spindle fibers from the centrioles. They are still in homologous pairs. During the anaphase, the spindle guides the movement of the chromosomes towards the poles, and the sister chromatids remain attached and move as an unit towards the same pole. Afterwards, homologous chromosomes move towards the opposite pole, and this is a difference to mitosis, because chromosomes appear as individuals instead of pairs. In the telophase, which is the end of the first meiotic cell division, the cytoplasm divides, forming 2 new daughter cells. Each of those newly formed cells has half the number of the parent cell chromosome, but each chromosome is already replicated, ready for the second meiotic cell division. In the prophase 2, which is similar to mitosis, but without having interphase 2, each of the daughter cells form a spindle and the double-stranded chromosomes move towards the equator. In the metaphase 2, chromosomes are positioned on the metaphase plate in a mitosis-like fashion, followed by the separation of sister chromatids in the anaphase 2. The sister chromatids of each pair move towards opposite poles, now individual chromosomes. During telophase 2 and subsequent cytokinesis, nuclei form at the opposite poles of the cell and cytokinesis occurs. After completion of cytokinesis, we have now our 4 daughter cells, and all of those daughter cells are haploid or have a chromosomal configuration of 1n. And again, the only cells having this chromosomal configuration in the human body are sperm cells and egg cells. If those cells fuse, we end up with a zygote, which has a diploid set of chromosomes and chromosomal configuration of 1n. To recapitulate what we have learned a bit about the meiosis, I'm gonna show you a quick video which summarizes this very nicely and includes fairly beautiful animations.
The cell prepares for meiosis just as it does for mitosis. But as the DNA condenses, each chromosome finds its homologous counterpart. These matching pairs of chromosomes hold equivalent genetic information, one set from mother and one from father. Next comes the first shuffle of nature's genetic deck. The 2 homologs trade genes in a process called crossing over. This forms new hybrid chromosomes. The spindles then place maternal and paternal chromosomes randomly on each side of midline. It is another shuffle that can lead to a host of different genetic outcomes. Finally, spindle fibers pull the homologs apart. This leaves the sister chromatids intact. With twice the amount of genetic information the cells will need, nature must cut the genetic deck in two. The second division produces 4 unique sets of chromosomes. Because they carry half the genetic information, we call these haploid cells. Many of them will mature into sperm and eggs, the reproductive cells called gametes.
So now that we understand how meiosis works, let us have a look at the consequences of errors during meiosis. And the first relatively common error of meiosis, it's so-called non-disjunction. And non-disjunction is actually leading to different genetic disorders, including trisomy 21. Here, a non-disjunction of chromosomes results in one sex cell which has 2 chromosomes of 21 and another one which has not a single copy. If those cells now fuse with a normal sex cell, the chromosomal configuration for this chromosome is actually 3. So we have one additional copy of the chromosomes because they haven't been equally distributed during meiosis. If the sex cell, which has no copy of this chromosome, fuses with a normal one, the result is a so-called monosomy, where only one copy of a certain chromosome is present. And this can indeed also lead to certain genetic disorders. And we are going to be looking at those genetic disorders in one of our future lectures. Again, I showed you this slide before. One typical example of a disorder resulting from non-disjunction in meiosis is trisomy 21, where we have 3 chromosomes number 21. What's also quite interesting is that in vitro fertilization drugs may be linked to genetic defects discovered in many embryos. This is actually quite common in terms of producing non-disjunctions. So what other errors in meiosis can lead to genetic disorders? We can have regions of homology exchange by non-homologous regions, and this actually happens from time to time if those DNA sequences are similar enough, and recombination can start although those regions are not related. This can cause chromosome abnormalities or deletions. With this, I would like to thank you very much for your attention. I'm closing the session on mitosis and meiosis, and I'll see you very soon. Thank you very much.